Hello, my name is Jordan Thompson. I am an undergraduate studying organic chemistry under the advisement of Dr. Nicholas Alzameda. And today I'll be talking about my investigation into amino acid and dipeptide inhibitors for botulism. Botulism is a neuroparalytic disease caused by the bacteria Clostridium botulinum. It secretes one of the deadliest neurotoxins known to man, with 7 different serotypes classified A through G, in which A is the deadliest and is the form in which our research targets. Both infants and adults can contract botulism. Infants can get it through the consumption of bacterial spores through honey, and adults can get it through inhalation, wound, laryngectic, as well as foodborne botulism. The neurotoxin will go in through the gut lumen until it hits the circulatory system, where it will then target the neurons of the host. Normal neurotransmission shows that vesicles will form around a neurotransmitter called acetylcholine. These vesicles will then go and coordinate through coordination with a snare complex to release this neurotransmitter into the synaptic cleft that will allow then for muscle contraction. Now, if one were to then be exposed to botulism, the neurotoxins will go into the neuron through receptor-mediated endocytosis, where the neurotoxin that's composed of a heavy and a light chain will then disassociate. The light chain will then go, and for serotype A, it will cleave a SNAP25 peptide that is a part of that snare complex. And therefore, it will no longer be able to release acetylcholine into that synaptic cleft and will then result in flaccid muscle paralysis. Now, this light chain is a zinc metalloprotease, in which the zinc atom is important as it is a cofactor that allows for this catalytic cleavage event. And in terms of botulism, there's no current treatment, but one is desperately needed due to a potential bioterrorism threat. Small molecule therapeutics are then a very viable option as they are cheap to produce. They're very stable in terms of not only oral administration, but also long storage times, and they can be mass produced and distributed in the event that a bioterrorism incident were to occur. So much like if someone were to throw a wrench into a machine to get it to stop functioning, one can also have the same principle applied for the light chain, where we can use small molecules to stop the catalytic cleavage event that occurs. And so the purpose of my research is to develop small molecule therapeutics to cure botulism. And in doing this, I'm following previous research that has shown that for the parent compound to the left, it has 4 key components to it. It has a tricyclic ring system, a sulfonamide linker, an amino acid core, as well as a hydroxymic acid, where this hydroxymic acid is very important because it allows for our inhibitors to chelate with the zinc atom in the bond light chain, and is something that we keep consistent for all of the scaffolds that we synthesize. So the first study that we did was to test the significance of the hydrophobicity of the parent compound by creating a bicyclic ring system from this tricyclic ring system. And furthermore, we wanted to probe different polar interactions by changing the positioning of the hydrogen bond accepting or oxygen molecule to the 3rd position of furan rather than the 2nd position. This is the compound that I synthesized, and in order to do so, the methods I used was a sulfonyl chloride coupling to create a methyl ester that I did a cross-coupling Suzuki reaction with to yield the following product that I hydrolyzed into a hydroxymic acid as my final compound. So the parent compound we used had 61% inhibition at 30 micromolars of inhibitor. And when we reduced the tricyclic to the bicyclic ring system, we saw a reduction in inhibition to 49%, which tells us that the environment of the active site is exactly hydrophobic as we were testing. And when we were testing the location of the oxygen positioning, we saw a further reduction in that bicyclic ring system to 14%, which signifies that the polar interactions are very specific to the location that they are positioned at in the fit of the inhibitor in the active site. The second study that we performed was a sulfonamide alkylation, in which the parent compound has a bent molecular shape. And this is caused by the tetrahedral orientation of the sulfonamide linker. What we wanted to do was explore the space above this compound by alkylating or adding on different side chains to that free nitrogen or hydrogen bond donating group to see whether or not we can probe further interactions to increase inhibition. This is the compound that I created, in which to do this, I did a sulfonyl chloride coupling, and then I reacted an alkyl halide to that free nitrogen to add a substituent onto the nitrogen. I did a Suzuki coupling and then hydrolyzed to create my hydroxamic acid. So when I was synthesizing this, I also wanted to test how the size of the alkyl chain affected inhibition. In doing so, I increased it from a methyl group to an alkyl chain and finally to a phenyl group. And in doing so, we saw that the percent inhibition reduced to 22% from 61% and further decreased the larger the side chain grew to no inhibition, which indicates that there were steric interactions occurring once we added to that free nitrogen, and therefore that is not a location that one would go about adding to when creating further therapeutics. The final study that my research lab conducted was a dipeptide core study in which we added on a second amino acid towards the N terminus of our inhibitor, and we decreased the size of our tricyclic ring system to a phenyl group. And then we also wanted to maintain those polar interactions that the benzofuran previously introduced by testing probing the positioning of the chlorine rings on the phenyl group. This is the compound that I synthesized, in which the two amino acids that we kept consistent was cyclohexyl as well as L-isoleucine. And then we introduced different chlorinated groups onto the phenyl group later in the study. To do this, we did a sulfonyl chloride coupling to create a methyl ester that we then hydrolyzed into a hydroxamic acid, which is more reactive and therefore could react with the second amino acid, L-isoleucine, to create a dipeptide methyl ester that we hydrolyzed into a hydroxamic acid. Now, what we found with this study was that when we created this 3,4-dichloryl dipeptide compound, the percent inhibition increased to 96% from the 61% the parent compound initially showed. And furthermore, when we tested a second dipeptide using 3,5-dichloryl phenyl group, we saw that the inhibition skyrocketed to 100%, which tells us that at 30% micromolar concentration of inhibitor, not only do we see good interactions and inhibition, but when we decrease the concentration of inhibitor to 100 nanomolar, we're still seeing activity with the second compound, which introduces the idea that it is very potent, and we wanted to probe those interactions. So we took the second compound and we docked it into the crystal structure of the bond light chain. And in doing this, we saw there was coordination of the hydroxamic acid with that zinc atom. We also saw that the L-isoleucine was protruding out of the active site while the sulfonamide was pointing into the active site. And furthermore, we saw that the cyclohexyl group was creating a plethora of hydrophobic interactions, which further provided evidence of the first study that we did. And the last component was the phenyl group with the two chlorine atoms attached, in which we saw that there was a variety of polar interactions as well, which further introduced the hydrogen bond accepting location significance from the first study. The key findings that we obtained were that not only was the hydrophobicity and location of the hydrogen bond accepting important and significance for inhibition, but also with the sulfonamide alkylation, we introduced the idea that these inhibitors' bent molecular shape is pivotal for inhibition. And furthermore, when we did the dipeptide study, we saw that increasing the scaffold then allowed for very potent inhibitors in different interactions. So by doing the structure-activity relationship, we were able to create an archive that future scientists can work off of when developing therapeutics, not only for zinc metalloproteases, but also for BONT itself. And finally, with the dipeptide study, we were able to see a potent enough inhibitor that we can create further analogs of and eventually go not only to in vivo studies, but also to clinical trials down the road. Thank you so much. I look forward to your questions.